Well, all right. Thank you for the opportunity to present here. Good evening to colleagues on the African continent and morning to my colleagues here in the state of Washington. Um, I'm Ken Thummel. I'm a professor in the Department of Pharmaceutics here in the School of Pharmacy at UW. I'm presenting today on behalf of my graduate student, Chris Arian, who I really wanted to do this presentation, but unfortunately he had other commitments and wasn't able to make it. And then also want to mention my colleague Ed Kelly here in the department, who's been instrumental in the development of the platform that I'm going to speak about. So this talk is a little bit of a departure from the first two you've heard. It is a spin-off project that evolved from our natural product drug interaction studies that the NAPD Center has conducted over the last 6-7 years. And in an effort to develop a new tool that might help us make these in vitro, in vivo predictions of natural product drug interactions. And so that's what I'm going to present. I have to say at the outset that this work is really in its infancy. It's technically demanding platform, and we've been learning along the way. So a lot of what I'm presenting is it's all unpublished, and it's really in its earliest early stages. But I think it's interesting enough, and hopefully you can appreciate the potential that this platform has for a lot of drug disposition research. So to start with, though, this, of course, the genesis was a natural product drug interaction that was conducted by Mary Payne and colleagues at WSU to evaluate the effects of the product Golden Seal on drug disposition. And if you don't know it, Golden Seal, it's an herbal supplement. It's been it's a plant that grows here in North America, southeastern Canada, and the eastern U.S. Been used traditionally by indigenous peoples in this country for long periods of time. And is a popular drug here in the U.S. Treatments for common cold, digestive diseases, and more recently we were aware of its use in type 2 diabetes. The two major constituents of Golden Seal that have this potential for drug interactions are berberine and hydroxysteine. These two molecules, they're the most abundant in Golden Seal. Phytochemicals, and they both contain what medicinal chemists would call a structural alert, this methylene dioxyphenyl moiety, and which it's been well documented can undergo biotransformation to generate a carbene that binds very strongly to the heme in a coordinate bond way to basically inactivate cytochrome P450s. And so that was our initial interest in this molecule, and there had been work to characterize it. But there was also the question of whether these molecules might also do things to other proteins involved in drug disposition, in particular the transporters. So we set out to study it more thoroughly. The initial work involved in looking at the ability of these molecules to inhibit P450s, work that Mac McDonald and Alan Reddy's lab led. And indeed, they are mechanism-based inhibitors with bioactivation of that methylene dioxyphenyl moiety to lead to inactivation of CYP3A and other P450s. But it was the CYP3A that we were most interested in because of its prominent expression in the GI tract and the intestinal mucosa. The PBPK model that was based on this in vitro work predicted potentially a fourfold increase in oral midazolam AUC, and in part because of the potential to inhibit intestinal CYP3A enzyme. But also there was simply the unanswered question as to whether the constituents of Golden Seal might also inhibit transporters, in particular transporters in the intestine, the liver, as well as the kidney. And so a screen was conducted to evaluate that potential for inhibition using cell systems that express various transporters here. And based on those results, it led to the following predictions here of a potential effect of golden seal on these transport functions. In particular, the data suggested that there might be changes in rosuvastatin as a consequence of changes in BCRP, as well as OATP 1B1 and 1B3 activities. But then it predicted that there wouldn't be any interactions with other transporters, in particular OATs and OCT and MATES. And so Mary and colleagues designed a study, the pharmacology core of the NAPD Center, to evaluate both the interaction with CYP3A in the gut, but the potential to inhibit the functions of various transporters. And so a study was conducted. I'm not going to go into it. It's published. You can look at the details there. It basically looked at the effect of 1 gram of golden seal 3 times a day for 5 days on the disposition of a cocktail of molecules, which included midazolam, furosemide, metformin, as well as rosuvastatin, and the results of that were actually somewhat surprising to the team. The first was the fact that there was no effect on rosuvastatin disposition. This was work that Josh and his team actually had led in the analysis of the samples that were collected from the interaction study, and there just simply was no evidence of a change in both systemic, the intestinal, as well as renal disposition of rosuvastatin, which is the 3 different PK parameters just shown here. And there was no change in furosemide as well, again, suggesting really no interactions in the kidney of kidney transporters. There was the interaction with midazolam, our CYP3A probe, which confirmed what had been published, and an increase in both the Cmax as well as the AUC, but no change to any great degree in the half-life, suggesting that this interaction occurs predominantly in the small intestine, affecting the intestinal bioavailability of midazolam. But the other interesting one was the interaction with metformin, and actually seeing a decrease in metformin AUC. And again, with data that suggested that, the lack of change in half-life, that the interaction involved intestine, in particular, the renal disposition of metformin was not altered, the renal clearance of it. So both of these results suggested an interaction that was predominantly in the intestine, and that wasn't well predicted by our models of working with human liver microsomes, as well as cell transporter expressing cells. The other thing I want to highlight was the magnitude of the midazolam interaction. It was less than that predicted by the in vitro, in vivo extrapolation from liver microsomal and intestinal microsomal experiments. And so that led us then to rethink this question of platforms, models for screening intestinal drug-drug and natural product drug interactions. And so key here, just to focus you, is on that intestinal disposition. Orally administered drugs obviously can transit the intestinal epithelial in a passive way. They may in fact be potentially pass paracellular, although that's very limited passageway, but they can also be transported in both across the apical and basolateral membrane. And then, of course, the efflux transporters potentially inhibit this. And then lastly, you can get metabolism within those enterocytes. So we decided we needed a better tool to evaluate both that disposition as well as natural product drug interactions that might be localized to this tissue. And if you've done any work in this space, you know that the systems that historically have been used, there are a variety of them, but they all have limitations. Many of them are fairly good at looking at permeability, but poor at looking at metabolism. KCO2, for example. Some, like LS180 cell lines, good at metabolism, but don't form the tight junction barrier, paracellular barrier. Primary enterocytes, you can look at activity, but only of a short duration, and you really can't look at transport activities. So this led us to really pursue alternative model. And what I'm going to talk about are the intestinal organoids. Not going to say much about MPS, but you can develop MPSs based on the organoid technology. And this technology evolved from a very seminal discovery in 2007 from Hans Kleber's group in Europe of identifying for the first time the intestinal crypt cells that give rise to the rest of the cells that populate the villi of the small intestine, as well as the rest of the regions of the intestine. And these are LGR5 positive crypt-based columnar cells. Staining here is the LGR5 positive, and you can see their localization down here in the crypts. And they did some really elegant studies to demonstrate that, in fact, these cells, as they replicate and then begin to migrate up the villus, in fact, give rise through differentiation to all the cells that populate the villus, and importantly, the enterocytes, which is the focus of our interest there. And additional work had led to the ability to isolate these cells just from pinch biopsies of the small intestine and other regions of the intestine. A fairly straightforward procedure for isolating these cells, and then by culturing them, in this case here on Matrigel plugs, they can be replicated to give you cell numbers then that can be useful for studying drug disposition processes. As they replicate, they give rise to what are called spheroids. They're all LGR5 positives. These are essentially undifferentiated cells. And our colleagues at Johns Hopkins, who really led us down this pathway, they use these to then allow them to differentiate through the removal of Wnt3a, a key growth factor, and they generate what are called enteroids, 3D enteroids. They have within this structure all the different cells that are found within the intestine, small intestine there, including the enterocytes. But importantly, they actually form a configuration where the tips of the quote villi are actually internal, and the crypts are external to the matrix on the external surface. And so it's an interesting model, and they use it to study a variety of diseases, in particular the impact of microbial infections on intestinal function. But for our purposes, they're not that useful, because one would have to, if one wanted to study oral disposition, do microinjection, which would be a tedious process and very, very challenging. And so we identified a method that allowed us to actually separate these spheroids into individual cells and then populate them on, to start with, a transwell culture platform. So essentially creating a monolayer of differentiated enteroid cells. In other words, starting with the undifferentiated, and then by changing, withdrawing, or reducing Wnt3a, they will begin to differentiate into the multicellular types that we are of interest. And so this would then allow us to have a model potentially with superior functions that could be used to study drug interactions and natural product interactions. Key for our interests is control of the Wnt3a concentration. Just shown here is a cross-section of the intestinal biopsy in the small intestine. And you can see here, obviously, the crypt cells originating down here. And as these cells migrate up and differentiate, shown here is the expression of CYP3A4 in brown. And so we wanted a system that replicated this expression here, as well as other functions, transport functions of these villus cells. And what we discovered, and had been alluded to by others, was that if you actually just simply reduce the Wnt3a and not remove it altogether, that you could get cells that were stable for prolonged periods of time. Whereas with complete withdrawal of Wnt3a leads to a basically apoptosis within a matter of 5 to 7 days. And so this gave us then potentially a platform that would be stable, but that would also allow expression of the drug disposition proteins of interest. So that's what we set out to do. Culturing them on the transwell. This is just bright field images of 2 donors here. And you can see that they will form confluent monolayer. Evidences of protuberances in villus to be confirmed by further microscopy and imaging. But key, what we wanted to determine was, in fact, were they differentiating with the reduction in Wnt3a? And this is just some imaging work looking at villain, a marker protein of differentiation, not found in the undifferentiated cells. As day one culturing, but clearly evident within the cells by day 7 here, the structural protein of villain in what looked like majority of enterocytes. In addition, another key imaging experiment was to look for evidence of the presence of stem cells within this population. Were there still stem cells because of the presence of some Wnt3a within there? And this is staining looking for the Ki67, which is another marker nuclear protein of rapid differentiation, or rapid replication rather, in undifferentiated cells. And indeed shown here was that the evidence of, in the nuclei of some of the cells, that they were indeed still stem cell phenotype with the biomarker of the ability to replicate dividing. So we think this is a potentially stable platform. We've cultured it out to 3 weeks that has some continual propagating cells and then differentiated cells that impart the functions that we are interested in: drug metabolism and transport. So what I want to show here is just our efforts to characterize that. Some of the data we've generated, first looking at permeability. We evaluated it early on, day 7, and then throughout a 3-week period. Shown here on the left are the TEER, the transepithelial resistance. It's just a marker, if you will, of the formation of tight junctions. And indeed, we see in 3 donors shown here, this formation, increase in TEER values consistent with the formation of tight junctions. If you culture undifferentiated cells out and keeping Wnt concentration high, you do not get that tight junction formation. So it clearly is a property of differentiation. And the TEER values themselves are similar to that, which you would get, at least in our hands, of CACO2, a bit lower than other cells, T84, and much higher, of course, than the LS180 cells, which we knew previously. Looking at probe permeability, paracellular probes, we studied luciferase yellow and atenolol. Indeed, we found that early on, these two probes, as well as midazolam, essentially our control, a highly permeable molecule, were all highly permeable at day 7. But by day 21, those tight junctions had formed, and indeed, the 2 molecules we wouldn't expect to passively perfuse the epithelial monolayer indeed didn't. Their permeability was greatly reduced. In contrast, midazolam permeability remained. So that gave us clear functional evidence that we had tight junction formation. Chris did some additional experiments looking for some of the tight junction proteins. This is just data looking at ZO3, one of the tight junction proteins. And indeed, you see that formation occurring that between cells here. Of the expression of ZO3 and its absence not shown here in the undifferentiated cells. We next turned our attention to CYP3A activity, again in experiments conducted using midazolam as our 3A activity probe, evaluating it baseline day 7 as well as day 21. And of course, our interest in 3A is the fact that it's the most abundant of the cytochrome P40 in the intestine. We evaluated it by measuring the midazolam extraction ratio across the epithelial barrier following an apical, or what would be modeling an oral administration, and then its disposition, its appearance in the basolateral compartment. Our extraction ratio is just the sum of all the metabolite form, 1-hydroxymidazolam, over the sum of the metabolite plus the midazolam that escapes into the basolateral compartment under sink conditions. We ensure that we still maintain sink conditions. And what's shown here is indeed we get very significant midazolam extraction and that it continues to increase between day 7 and day 21. It's inducible by vitamin D treatment, which consistent with work we had done previously on vitamin D effects on CYP3A expression. And then there was inter-individual variability. Some donor cells showed very significant high extraction ratios, others a little bit less, but both of these extraction ratios, 20 to 45%, very consistent with what we had observed, Mary Payne had observed in vivo. Last data I want to just show, we've got others, is just PGP activity. We evaluated again at day 7 and day 21 with digoxin, and evaluated under basal conditions, rifampin induction, and verapamil inhibition. And we demonstrated this by looking at the basolateral to apical flux permeability of digoxin compared to the apical to basolateral flux. And what you can see here is indeed what you would expect with the PGP-mediated efflux activity is higher permeability of digoxin in the basolateral to apical compared to the apical to basolateral with an efflux ratio of 3.6. That increased a little bit with rifampin treatment, and it declined with verapamil treatment, in particular, a decline in the basolateral to apical flux. So this gave us evidence that these cell monolayers were also developing PGP-mediated efflux activity. So the conclusions we have, we've got a lot of more work to do, but we clearly have a robust system, robust inducible 3A activity, given its extraction ratios is what we would expect in vivo, high barrier integrity and tight junctions forming, and evidence that we're getting the transport activity, although we do need to do a lot more work, particularly with the OCTs and other transporters that might be mediating metformin disposition in the GI tract. And so our hope is that this model will allow us to probe more thoroughly and mechanistically the potential interactions of natural products and drugs in the intestine, which, if you think about conceptually, are highly likely given the high levels of exposure of natural product phytochemicals to the GI epithelial cells. So with that, I'm going to stop and acknowledge a lot of people, of course, our collaborators who are part of the Golden Seal study. Mark Donowitz at Hopkins, who kindly gave us the initial cells, donor cells, stem cells that we've been working with. All this work has been done by Chris Arian, as I measured, but supported by others in the lab, as well as the PK lab. The early work that Alan and Matt McDonald did with the mechanism-based inhibition, and then colleagues in the Kelly lab as well as Young lab. And a lot of funding. Chris has been fortunate to get funding for it, but in particular, the funding of the NAPD Center. So I'll stop with that and take questions from others.
Thank you, Ken. This is such exciting work. We have needed an intestinal model so desperately for a long time. So I'm glad it's you that's working on the technical details of it, but we really look forward to having this model accessible someday.
Yep.
So are there any questions? I don't see a question yet in the chat, but have you published it all on this yet, Ken, or you're just
no,
not
we haven't.
Yeah,
we want to do that, and Chris is working diligently on kind of filling in the gaps of what would be the first publication on just general characterization. Obviously, we're not going to get everything we would like in that, but we feel compelled to get something out there quickly. We know there are a lot of other groups working in this space, in particular colleagues in the pharmaceutical industry. My suspicion, we know of some, but my suspicion is many companies are working on this platform. And of course, there are commercial platforms out there that are getting marketed as well. But I think everybody has come to the realization that this is as you alluded to, the direction forward. And I think it's just going to be a really useful platform for natural product drug interactions. Yeah,
absolutely. Absolutely. Okay, I don't see any questions yet, so we'll continue to monitor, but I think our next speaker, Kumbhakani, is next. So if you want to share your slides.